Okay, so I think that today's webinar has started. Thanks to everybody for joining. And this time we may even have some colleagues from the US because our speaker, Alex MacKerell, is from the US. Thanks a lot to him for accepting to give a talk. So I will start by introducing Alex a little bit. Alex is, Alex MacKerell is professor of pharmaceutical sciences at the School of Pharmacy, University of Maryland. So that's sunny Baltimore on the East Coast. And many years ago, Alex started, in fact, as doing mostly experimental work. His initial training, I think, was mostly in biochemistry. He did his PhD at Rutgers University and then continued as a postdoc at Karolinska Institute in Sweden. And that's where he started to develop an interest in computational methods. So then Alex got further training in computational methods at Harvard University, working in Martin Karpler's group, who developed the well-known CHARM package. And indeed, Alex essentially took it essentially took the responsibility, largely de facto, to develop the CHARM force field, which is used by many people to simulate biomolecules, but also small organic molecules. And this, of course, ties well with, is essential for his current interest in drug discovery. So among his responsibilities, Alex is also director of the Center of the Computer-Aided Drug Design Center at the University of Maryland. And another thing that Alex has done is to co-found and be chief scientific officer of the company called SilcsBio. And I think SilcsBio, or its underlying science and technology, are the topic of today's talk. And you can see the title of the talk on the slide, so I'm not going to reread that, or Alex abstract, in fact. So we have I think enough time for us to answer questions at the end of the talk. So if you have questions, please write them in the Q&A panel at the bottom of the Zoom window. I think without further ado, I will let Alex give his presentation. Thanks again for doing that.
All right, great. Thank you, Nikolai, for the nice introduction, Sarah, for the invitation. And I will speak today about the SILCS technology, which we've developed probably initially in like 2008, came out first sort of like seed of SILCS. And then we've developed it in a lot of ways that I never anticipated it would go. So I'll give an introduction to sort of the concepts behind the technology, some details of like how you can start to use it for drug design. And then at the end, I'm going to sort of jump around with a number of more recent ways that we can apply the technology, which I think is important because it's not just sort of a very specific technology for like one use. It's actually quite broad. The basic concepts behind it are quite broad. We can use it for a lot of different things. Here's my conflict of interest side. And again, Nikolai said I'm involved with SilcsBio and some other ratio therapeutics. And then we have funding from the NIH and so on. All right, so the majority of you are familiar with computer-aided drug design. And obviously in computer-aided drug design, we want to sort of qualitatively think about how to make molecules better. Okay, so we've got to, can they bind better to the target protein? They've got to be synthetically accessible. We've got to consider bioavailability and so on. And we then want to then make a quantitative evaluation of like, will that ligand actually bind better, be more specific? And we want to be able to make to gain that quantitative information at a rate that can lead the design process. So the medicinal chemistry program can synthesize and evaluate, say, 100 compounds per week. Then our CAD methodology needs to evaluate, at least a factor of 10 more than that on a weekly basis. So we need a method that is of sufficient accuracy and speed that it can sort of lead the drug design process. So now, basically, if we want to do quantitative analysis, we've got to predict the binding affinity of ligands to the target protein or RNA or whatever. And here's the relationship of the concentration of a protein ligand complex to the absolute binding free energy. And obviously, this requires determination of the equilibrium between the bound and the unbound states, which experimentally, this can be done in a variety of ways. Computationally, we can do that using molecular dynamic simulation. And I have my little image of, integrating Newton's equation of motion for different time steps. However, to sort of really calculate this equilibrium using MD alone, it takes simulations in the order of tens of microseconds or more. So basically, this is obviously computationally possible, but very challenging and not really going to get us to be of utility in the concept of a drug design and medicinal chemistry campaign. So the way we've been getting around this is calculation of the change in the binding affinity as we modify ligands and so on. And this gets us to the free energy perturbation technology, which there are a number of different ways to perform free energy perturbation. I've got the examples of the exponential formula, the thermodynamic perturbation formula, and then the Bennett's acceptance ratio, which is used more, has been widely used more recently due to the ability to do the iterative solution and so on. Now, these methods, which are just getting the change in the binding affinity, are computationally more efficient than calculating the absolute binding affinity. However, you can see that in all of these equations, we've got our brackets here, okay? And our brackets represent an ensemble of conformations. So when we're doing free energy perturbation, for every new ligand, we have to generate this ensemble of conformations. And this is still expensive. And this can be done, say, overnight on a collection of, say, GPUs, but you're still limited to, say, 10 perturbations today on the computational resources most people have. And you start to think of like your software has to support doing a number of perturbations. You have the idea of tokens and these types of things. So this generating an ensemble of conformations for every new ligand is a giant rate determining step. So with the SILCS technology, we're now sort of getting to the idea of like a precomputed ensemble. So we're going to overcome the computational demands of calculating an ensemble of conformations for each ligand by pre-computing an ensemble of functional group distributions. We're going to do that once, okay? And then we're going to use that pre-computed ensemble for every new ligand. So we're going to do one upfront expensive computation, get this pre-computed ensemble of conformations, which we're going to refer to as SILCS FragMaps, then use those over and over again to calculate absolute binding affinities or changes in binding affinity and so on. And this allows us to literally do thousands of calculations, ligands on a daily basis. And this led to our this site identification by ligand competitive saturation SILCS technology. And I'm not really going to go into the CGNFF program. I've got one slide at the end. But the CGNFF program's important because if you're doing thousands of ligands, you need a tool that allows you to quickly read in ligands, generate the topologies, assign the parameters, and then do your calculations. And the CGNFF program does that quite nicely. It's basically you generate your molecules in no time at all. Okay, so now the SILCS technology. Basically, we're going to get a rigorous free energy evaluation of the functional group affinity pattern in the entire three-dimensional space in and around a protein. So we're going to use a combination of GC, MC, MD simulations. And these simulations include protein flexibility. We have explicit solvent representation. as well as an explicit solute representation. So we're including desolvation contributions, obviously protein functional group interactions, as well as protein flexibility in these free energy functional group affinity patterns, which we're going to refer to as FragMaps. And again, this is our precomputed ensemble. Once we have that, we're going to use that ensemble over and over again. And so basically what we're doing here, we can show it diagrammatically, is if we take some solutes and we put them in a box of water, and we do an extended MD simulation, and then we calculate the probability distribution of that solute throughout the simulation system, we're obviously going to get a homogeneous distribution. So then we add in a protein, target RNA, whatever, into the simulation system. And so now we get a probability distribution where we have higher probabilities in favorable binding sites for the solutes that we're looking at. We have lower probabilities in the regions which are unfavorable, obviously on top of the protein, but in pockets where a certain type of functional group doesn't like to bind. And then away from the protein, we have a probability which is equivalent to that in solution. So what we do is we take the probability distribution in the presence of our protein. We then normalize that for the probability distribution in its absence, which is just related to the concentration of your solutes. And we come up with this normalized probability distribution where we have probabilities greater than 1 in favorable binding regions, less than 1 in unfavorable regions, and then values of 1 out in solution and so on. So this is now the sort of the foundation of our pre-computed FragMaps. Now, when we do these SilCS simulations, we include a collection of 8 solutes simultaneously. So this is obviously greatly increases the computational efficiency. And you can see we've got aromatic, aliphatic, various hydrogen bond donors and acceptors, imidazole representing like a heteroaromatic, and then we've got negatively charged acetate, positively charged methylammonium, and so on. So we have this collection of solutes at a concentration of approximately 0.25 molar in the presence of an explicit solvent representation, and we do an extensive amount of sampling, approaching a microsecond of MD. And then from this, we can get the functional group probability distributions for all these different classes of functional groups and so on. We've extended the technology to do like a second class of solutes, halogen-containing compounds, which are obviously very important for drug design and so on. So we can have halogen maps in addition to our standard sets of maps and so on. So basically, we take our protein, we put it into basically a soup of water and these different 8 solutes. We then perform comprehensive molecular simulations. We then do a binning process into voxels. And from this, we now get these 3-dimensional probability distributions of the various classes of functional groups in and around the entire protein. So right away, we're not just focused on one binding site, we're getting the maps for the entire system. So this allows us to quickly go from just focusing on the binding pocket to actually starting to identify novel binding sites, all these types of things and so on. So we now have these FragMaps, and what we're now showing here are like FragMaps at high probabilities. So these are favorable binding regions for different classes of like apolar groups, negatively charged groups, donors, acceptors, and so on. Now, when we initially were doing SilCS, we're just applying MD simulations. And this, for the initial systems, we worked on this work quite well, but then you start to look at more challenging systems, such as GPCRs, where you have very deep or totally inaccessible binding pockets. And in here, I'm looking at the mu-opioid receptor with the agonist BU72 bound. This is just the crystal structure, and you can see that the ligand is fairly deep in the binding pocket. And if you look down from the top of the solvent accessible surface, it's largely inaccessible. But for the SilCS technology to be useful, we have to map out this interior binding pocket, as well as binding pockets on the surface. And this is quite challenging for just MD simulations alone, because basically our solute sampling is limited by the rate of diffusions of the solutes. And one of the things that we've done to overcome that is use small solutes. If you looked at the benzene, the propane, and so on, those are small solutes, which maximizes their diffusion rate. However, diffusing in and out of deep pockets a number of times is required to get a converged probability distribution is difficult. And then we also have the second problem of if the pocket's protected and totally has to open, we have to take into account the time scales of protein conformational changes. So we have these two challenges which we have to overcome if we want to get the precomputed ensemble throughout the entire protein system. And here I just sort of give an example of the androgen receptor, which is a nuclear receptor, where the binding pocket is actually totally occluded from the surface of the protein. The protein basically has to partially unfold for the ligands to bind and then refold and so on. So the way to get around this was like, okay, we can start to think about using the Grand-Canonical ensemble. And the Grand-Canonical ensemble we're simulating in the context of the chemical potential, constant volume, and constant temperature. So we actually can change the number of particles in the system. And we're doing this through insertions and deletions from external baths into the simulation system. And the acceptance of these insertions and deletions, as well as rotations and translations, is based on the Monte Carlo. We use Monte Carlo for the insertions and all the moves, and then use the metropolis criteria for acceptance based on the excess chemical potential for the solutes being inserted and so on. And the nice thing about this is now we can now get insertions and deletions, say, into the deep pockets that are totally inaccessible. So the Grand-Canonical ensemble helps facilitate simulation, sampling throughout the entire simulation system, but most noticeably, it allows us to do insertions and deletions into these deep pockets. So we don't have to worry about diffusion in and out, opening of the protein, and so on. And basically, one of the problems with this, though, is that insertions of large solutes, such as benzene, acetate, and so on, into a condensed phase environment, you have very, very low acceptance ratios. So what we did to overcome this is we've developed the oscillating excess chemical potential approach, where basically what we're doing is we're targeting a given concentration, and if the concentration is of the solute is too low, we actually turn down the chemical potential, which actually forces acceptance when we do insertions. And then if the concentration gets too high, we change the chemical potential to make the probability of acceptance of insertions low, okay? So basically what we're doing is we calculate the concentration of various solutes, we then change the chemical potential accordingly to that concentration. We then, for say 200,000 MC steps, we do Grand-Canonical Monte Carlo. We then stop again. We then reevaluate the concentrations, and then we change the chemical potential accordingly. So this gives us our oscillations. So for any sort of one chunk of GCMC, we have fixed chemical potentials, but then we adjust the chemical potential in between each round of GCMC to allow us to get appropriate acceptance ratios for insertions and so on. So this is oscillating chemical potential approach. Now this allows us to apply the SILCS technology to these deep and totally occluded pockets and so on. So basically, the SILCS protocol typically involves these GCMC MD iterations, where we do about 200,000 steps of GCMC. To resample are both the solutes and water distributions. We follow this by some short equilibration of MD and then 1 nanosecond of MD. So we're now allowing the solutes, the water, to getting additional sampling of the solutes and water. But now we're also bringing into account conformational flexibility of the protein during the MD simulation. We then repeat this cycle for 100 times, which gives us a 100 nanoseconds of MD sampling. And we do this for 10 individual systems. So we end up with a total of 1 microsecond of MD, from which we calculate the probability distributions, which are our precomputed ensemble, which gives us our FragMaps, which we now have for drug design. And basically, this calculation is about 3 to 4 days for a GPCR with a bilayer using 10 2080 GPUs. So we can now start to look at sort of some of the information content we're getting. Now I'm using as an example system, just protein tyrosine phosphatase, where you can now look at the distribution of the maps. We're encompassing the entire three-dimensional structure of the protein. Here's the binding site in here. And you can see your various hydrogen bond acceptor, donor, aliphatic, aromatic maps, and so on. What's interesting is you start to see how some of these maps start to overlap. So you can see obviously regions for negatively charged groups, positively charged groups, but you can see a region here, which is going to be appropriate for both aliphatic groups as well as hydrogen bond donors and acceptors. So your three-dimensional information is very detailed. You can say it's not just this functional group here, that functional group there. You can see regions where you can have multiple types of functional groups, all favorable for that region, and so on. Now we can look into the active site of the phosphatase. And now what I've done is I've just taken a ligand from a crystal structure and overlaid it on the maps. And this is just the crystallographic orientation. You can see how the maps correspond nicely to the different types of functional groups, the various aromatic groups, the donors and acceptors, and so on. So you can nicely see how the frag maps, these functional group distributions, nicely correspond to the expected locations corresponding to the functional groups on the maps. And now we're looking at the solvent accessible surface of the protein, which is based on the crystal structure. And now you can see here is how the frag maps actually start to penetrate underneath the surface. And this is because we're including protein conformational flexibility when we're calculating these maps. And we can see that more clearly here in this slide, where it's basically the same image to the left with the solvent accessible surface. And now to the right, we now have the same collection of frag maps, but we're now showing what we call the exclusion maps. And the exclusion maps are the forbidden regions where the solutes and the water never sampled during these SILCS simulations. So this is giving you an idea of the full extent that the protein can open to allow, say, a ligand to bind and so on. So basically, once we start to use the frag maps for ligand design, we basically get rid of the protein. We're then working in the context of the frag maps themselves along with the exclusion maps. And you can quickly see how much the protein can open to allow us to start to do our ligand design basically underneath the surface of the protein. So we get away from the limitations of a crystal structure per se, because we're now accounting for the protein flexibility and so on. Now, the method includes the explicit representation of water molecules. So basically from the SILCS simulation, we can also get frag maps for water. And what I'm showing here are the frag maps for water for factor XA. And what I've done is now, we did the SILCS simulation starting with the protein structure 1FJS. I now put the water positions from the second crystal structure of factor XA. You can see how the maps correspond well to the water positions from the alternate crystal structure. So you can start to see, we can see where there are water molecules which are going to be favorably bound. And what we can now do in the right panel is we now bring in the frag maps. So you can now start to look at the frag maps for the water molecules as well as the frag maps for the different types of functional groups. And you can start to identify regions where frag maps for various functional groups correspond to the frag maps for water. These are water molecules that can be displaced, okay? And you see other water molecules where you don't see the frag maps for other solutes. These are the water molecules that are going to be difficult to displace. So you have all this information about water, how to displace those waters. You can actually identify which types of functional groups would be best to displace those waters and so on. So again, this is just giving you an idea of the huge amount of information content that are in these frag maps once we have them. Now, that's sort of qualitative information. Now we want to move into the quantitative realm. And what I'm showing here is now basically what we do is now we take these normalized frag maps. We've normalized them from the presence of the protein by in solution. So again, basically, if we're out in solution, we have a value of 1 for this ratio. we take a log of that, we're going to get what we call the grid-free energy. So basically, a grid-free energy is now giving us information on a functional group's affinity relative to being in solution. So if the GFE value is 0, that means that the energy for that functional group, it's the same as it being in solution. A negative value says it wants to be, it's more favorable than being in solution. And a positive value indicates that it's unfavorable relative to the solution. That functional group at that location would rather be out in solution. So now we can start to look at our FragMaps at different energetic cutoff levels. You can see we look at the negative 0.5 versus negative 1.5. And so you can sort of really focus in on the more favorable regions where a functional group wants to be located, or more generally where that type of functional group can be. So we're now sort of moving in the quantitative realm. And so basically now we've got these three-dimensional GFE probability, GFE distributions, basically on a 1 angstrom grid. So now this allows us to now start to estimate the binding affinity for ligands. So what we're going to do is we're going to take each ligand and we overlay it onto the FragMaps. It could be just in the crystal orientation as we just saw. We then assign each ligand atom to a certain FragMap type. And based on the overlap of that atom with the FragMap type, we assign that atom the GFE score for that position in space. We then do a summation over all of the GFE scores for all of the classified atoms in each molecule, and we now get what we call a ligand grid-free energy. And this is basically our surrogate for the binding affinity. And basically, since we've already calculated these GFE FragMaps, calculation of this ligand grid-free energy is basically instantaneous. So you can now see how we can now use these pre-computed FragMaps very rapidly for ligand design and so on. So, and now once we've got these ligand grid-free energies, we can now go back and we can start to apply Monte Carlo sampling of the ligand conformation in the field of the FragMaps. So we can now do sampling of just say, we can overlay the ligand and say from a known conformation, do local sampling. But we can also take the ligand and we can do what we call exhaustive sampling or full-scale docking, where we do full rotations and translations of the ligand, as well as the ligand conformational degrees of freedom. So we can basically start from just, we have no known bound conformation. We can use our SILCS MC in the field of the FragMaps to identify bound conformations, assign them free energies of binding, and so on. So we can now, you can start to see how we can start to move into like a full quantitative use of the SILCS technology. And once we have that, we can start to do various correlation analysis. This is a study against the that we performed a number of years ago against the BCL6 protein, where we're correlating with, this is some binding data based on alpha lysa, and then some NMR, the chemical shift perturbation data, and so on. So now you can start to do your correlation analysis, starting to make predictions of how various modifications can improve binding affinity and so on. And now we can now sort of predict these changes in the free energy of binding, okay? But we also now have a very detailed picture of which atoms in the molecule are contributing to that, because we have the information for each atom. So here we have 2 molecules, which are from a congeneric series. And basically, we've taken a methoxide and added a sulfonamide. Now, the overall change in the binding affinity was a little over like a kilocalorie. But what's interesting here is you look at, we go from the methoxy to the sulfonamide, and you see that the change of that functional group is very large. It's like almost a 3.5 kilocalorie change. But the overall change is only a little over 1 kilocalorie. So what this shows is if we look at the rest of the changes, we look at the free energy contribution of the cyclohexyl group, it goes from negative 1.4 to negative 0.24. So where we've gained a lot of binding affinity, we've lost it in other portions of the molecule. So this is very detailed information that you can use to make decisions about where to go in your ligand design. So basically, you can start to see how this sulfonamide, we're getting much more favorable interactions, but that's sort of probably pulling the rest of the molecule out of its favorable interactions. And so where we gain binding, we lose it on the other end of the molecule. So now this is like, okay, we increase linker length, these types of things. So you have this very high level detailed information that can be used to facilitate your ligand design, which is basically inaccessible to most other computational technologies. Now, we can do the correlation analysis. I'll show you in a few slides how SilCS is very competitive with full-blown free energy perturbation. But what's interesting with the SilCS FragMaps is once we have the FragMaps, we start to have some experimental data, we can now actually optimize those FragMaps using a Bayesian machine learning approach, where we can actually improve the predictability of the SilCS LGFE scores. So basically what we can do is we can basically, we have our FragMaps as priors. As we'll see, those give us a very nice predictability. And then once we get some experimental data, even as few as sort of 10 experimental data points, because we have such a very good initial guess from the FragMaps, we can do a low data machine learning optimization to improve the predictability of the FragMaps with respect to these LGFE scores. And we can, as a drug design campaign proceeds, we can continually re-optimize the FragMaps to increase their predictability. As you have different congeneric series, we may optimize the FragMaps for one congeneric series versus another. But this is a very powerful way to continually improve the predictability of your model, but based on a very good starting guess. And this is important. These FragMaps are actually very predictable unto themselves, where we can do this optimization. And just to give you an idea of the extent of optimization, it's not huge. If you look at the scale here, this is like % correct. This is from 0.5 to 1. And here's the values from the FragMaps alone, and then we can increase their predictability. Not by a huge amount, but it gradually increases the predictability. And we can do this targeting % correct, correlation coefficient, RMS error versus the experimental data and so on. So this is a way that we can continually improve the predictability of the FragMaps. Again, it's something that's not accessible to like other types of methods. So now how do we stand up to other types of methods? So we're going to do a comparison of SilCS versus free energy perturbation. This was published a couple of years ago. And what we did is we used the 8 targets and the 199 compounds from the study by Schrodinger et al., which is something, this data set was subsequently used by a number of other labs using FEP. So you've got this collection of proteins, this number of ligands. And so basically, we're now using SilCS to get the relative binding affinities, comparing it to the published FEP data and so on. And so with the SilCS, what we do now is we've got basically our default SilCS methodology, which then gives us these metrics over all 8 targets and the 199 ligands. You can see the mean unsigned error, the R value, and the % correct. And then we can compare that to the various FEP methods. And you can see those numbers are quite comparable. So the SilCS technology, which is now, remember, this is like 100,000 times faster once you've got the maps calculated than the FEP, is quantitatively very comparable. We can now go in, instead of just using the default SilCS technology, we can select the best method, which is using, say, just the standard FragMaps or the standard plus the halogen FragMaps and so on, which increases our the predictability. And then we do this, the Bayesian machine learning optimization, and we move our predictability to be improves further. And you can see at this point, it's better than the various FEP approaches. And these again are from a number of different labs using various force fields, sampling technologies, and so on. So basically, the SilCS technology is hugely faster than FEP and is quantitatively comparable. And we were able to take the all the ligands, all 407 ligands, whereas the FEP studies only applied it to 199. And we can see that our SilCS technology maintains the predictability over all 407 ligands. And why the FEP studies only did 199 ligands. I was told it's because FEP is slow. I'm not sure about that, but so you can see the technology is quantitatively competitive with FEP, but thousands of times faster. Plus, you get this information on the contribution of individual atoms, functional groups to the overall changes in binding affinity. And this is just the workflow, but down here, I want to just emphasize a couple of points. The calculations that we did with SILCS, we did not start with a known binding orientation, okay? We did the docking of full three-dimensional sampling of rotational, translational space, as well as conformational space of ligand. So the SILCS technology gives us FEP quality accuracy, but we don't need to know the initial orientation of, say, the lead compound. The methodology is accessible to large chemical modifications, adding full-blown rings and so on. Basically, it's hundreds of times faster. We've made further improvements than FEP. And also, the impact of the force field is minimal because your free energy is based on the overlap of the type of atom with the frag map, not based on subtleties of the partial atomic charges and these types of things. And this in some ways sort of kills me to say this because I'm a force field guy, but it's actually sort of a lot of the limitations in force fields are not having as large an impact in the SILCS technology versus FEP, Poisson-Boltzmann, these types of things and so on. So I can now just go into sort of an example case, and this is actually from a number of years ago, where we applied SILCS to the beta-2 adrenergic receptor. And the goal here was to identify novel agonists, which could be potentially used for the treatment of asthma and so on. And what we did is we took advantage of the fact that the crystal structures of both the active and the inactive form. So we generated the SILCS FragMaps for both conformations. And now what we can do is we can actually start to look at difference FragMaps. So we can use the FragMaps to sort of both qualitatively and quantitatively understand differences in binding to the active state versus the inactive state. We're then going to take that information. We're then going to develop pharmacophores using these SILCS FragMaps for the active form to do screening in a large database. And then we're going to, from the pharmacophore screening, we're going to use both, in this study, we used AutoDoc, followed by SILCS LGFE scores to select some agonists for experimental evaluation. And so here we can look at the FragMaps in the active conformation, the inactive conformation. Now we're in the orthosteric site. And we can now look at different FragMaps. So what we can start to see right away is, in a qualitative sense, which regions are important for driving agonist versus antagonist binding and so on. So again, these difference FragMaps are going to be very powerful to, again, understand agonism versus antagonism. But you can also use it to sort of understand specificity for, say, two different kinases. I've got a quick example where we've applied it to some of the BCL2 proteins and so on. So what we now do is we can now we did a validation. And what we have here is correlation analysis using the active FragMaps and the inactive maps with a collection of agonists, partial agonists, and antagonists and inverse agonists. And what you can see is we get better correlation with the agonist, with the active maps versus the inactive maps. And when we go down to the antagonist, inverse agonist, better correlation with the inactive maps versus the active maps. So you can see that we're going from just sort of this qualitative picture of the difference in the active state versus the inactive state to some quantitative differences and so on. So this is going to facilitate using this sort of information content for the rational selection of compounds which are agonists. So what we've done is we've now taken the FragMaps from the active form. We've converted them to pharmacophores. We've got an automated method to do this. We then took these pharmacophores and used them for screening through large databases. Obviously, now we can go to the pharmacophore screening is very fast. You can go to, we've got like enamine set up for like hundreds of millions of compounds. with multiple conformations. We can do our pharmacophore screening based on RMSD. With respect to the pharmacophore, we then select a large number of compounds. We then do select compounds based on energetic difference criteria. We initially selected 1,000 compounds using AutoDoc Vena. We then selected a subset of those compounds using the SilcsBio LGFE scores, from which we selected about 100 compounds. Of those 100, we selected 15 compounds based on chemical diversity. And our experimental collaborators then took those and looked at activation in an ex vivo intact airway smooth muscle experiment. And what we found is that 7 of the 15 compounds tested were indeed agonists, and comparing to isoproterenol, which is sort of currently on the market for asthma treatment and so on. So basically, the methodology was quite successful, where we can actually sort of differentiate between agonists and antagonists and use this in it for database screening. And obviously, you can see how you can start to use this for your ligand optimization and so on. And again, here's this other study that I referred to, trying to develop specific inhibitors for MCL1 over BCL-XL. And again, we can just have FragMaps for the 2 proteins. We can get LGFE scores in the presence of the 2 proteins. We can then do the correlation analysis between the differences in the LGFE predicted binding affinities versus the experimental differences. And you can see the predictability is quite good. And we can then now move that to now what I'm showing here is the different contributions of individual atoms for binding to BCL-XL versus MCL1. So we can actually start to understand which atoms in the molecule are contributing to the differential binding. And again, this is going to be important for the design of specific inhibitors or the design of inhibitors that would bind to both proteins and so on. And so you just have this very detailed information. Okay, so now I'm going to sort of, I could say quarter of, I'm going to go through some various alternate uses of the SilcsBio technology. And because we have the SilcsBio FragMaps for the entire 3-dimensional structure of the protein, we can start to use it to identify novel binding sites. So we've applied this to a number of targets in a published study, where you basically go in, you do your Silk site simulations, you get the FragMaps for the entire 3-dimensional protein. What we do is we then do a comprehensive SilcsMC docking of fragments into the entire 3-dimensional space of the protein. And we typically use a set of approximately 100 mono or bicyclic rings. There's also this set of compounds, these called mini frags from Aztecs. And what you do is you're basically taking each ligand and doing this full docking, tens of thousands of times in the full 3-dimensional space of the protein. We then get these thousands and thousands of docked orientations of each of the small fragments. We go through 2 rounds of clustering for the individual fragments and then for overall fragments, from which we identify these, what we call hotspots. Then once you've got these hotspots, what you can then do is sort of we identify hotspots which are, say, 2 or more hotspots adjacent to each other, which would mean that these smaller fragments could be linked to make drug-like molecules. And then more recently, we didn't do it in this initial study, but we actually go in and we then, for each of these regions that we think that look interesting as a novel binding site, we basically do a docking of a set of about 350 FDA-approved compounds, which are drug-like, and then evaluate the sites for actually based on the predicted binding affinity as well as sort of the relative solvent accessibility to identify those sites which would be most appropriate for the drug binding. And we've used this to identify a number of allosteric sites. We identified some sites on glucosidase A, where glucose 6-phosphate binds, and these were experimentally validated. In another study on heme oxygenase, This is interesting because the ligand design was actually targeting the heme binding site. And we went in and we did the SILCS calculations, and the results, the experimental analysis, especially some hydrogen deuterium exchange, was like, it's not binding to where we think we are. We were able to identify a site on the back of the protein. Again, this was from the initial maps. We didn't have to recalculate the maps. And then we were able to show that the binding affinity of the ligands was typically more favorable to that site than to the heme binding site. And so we basically identified this allosteric site. And then the hydrogen deuterium exchange validated was consistent with the HDX data and so on. And so we're getting short on time, but we were also able to understand what was interesting with this the system is we had very flat SAR. The compounds went from a hydrogen to like a naphthyl group and so on, but the affinity didn't change very much. The EC50 in antimicrobial experiments changed more, and we're able to use the LGFE contributions and the GFE contributions for individual functional groups to basically understand why we're getting the flat SAR. And again, it was sort of this push-pull mechanism where we sort of gained binding on one end of the molecule, we lost it on the other end, and so on. So again, it's that information concept. And then more recently, again, we got back to the beta-2 adrenergic receptor. We have an experimental collaborator, and we were actually used the SILCS to again go in, identify a totally novel allosteric site. We used the SILCS pharmacophore screening and the SILCS MC docking to then identify ligands for that site. And we actually identified both allosteric activators and negative allosteric modulators for that site, and which was experimentally validated and so on. And that's a more recent publication that came out last year. So we can use it for allosteric sites. It's very good, and because we're keeping this conformational flexibility of the protein. So sites that are not in the crystal structure, you can ultimately see them. And this is very powerful. We've extended the technology to, if you want us to target small molecules against RNA. And basically you have the same sort of information content as you have with proteins, the maps, identifying where different functional groups bind, using it to identify novel sites. But RNA is really tough. And I know Nikolai knows this, as this is just examples of the different structures of the HIV TAR RNA in the presence of small, different small molecules. See, the overall structure is the same, but these subtle changes, it tends to be like sort of crankshaft motions where different pockets open in different ways. And this makes RNA very, very difficult to study. So SILCS is helpful for RNA. I can't say that we've solved all these problems yet, but it's something to think about if you're interested in RNA. Recently, we've extended the methodology for covalent drug design. At this point, we've done it to in the context of targeting cysteines. And basically what we can do is we take our SILCS FragMaps. We also now calculate the probability distribution of the cysteine sulfur. So we have the inherent space being sampled by the sulfur. And basically we can now start to do ligands of our warhead molecules into the SILCS FragMaps in the vicinity of the targeted cysteine. And we can start to understand the binding affinity adjacent to the targeted cysteine, binding affinity in reactive conformations where it overlaps with the cysteine, and we can then start to use that to identify potentially reactive cysteines in a protein. We then we use a database of 24 known warheads, and we then combine that with AI to develop a predictive model to predict which specific types of reactive functional groups will be useful for targeting with a cysteine. And we can show how we can now use our grid-free energy information to sort of optimize the non-covalent scaffold interactions with the protein, is what you would need to sort of improve the, say, the specificity of your covalent agent and so on. So you can see here, now this sort of gets to a theme that we're focusing on more these days, is sort of taking the underlying physical model of the SILCS FragMaps and then combining that with machine learning for that Bayesian optimization. You saw that with now developing an AI model against known experimental data for known warheads, and then combining the physical model with some learning to make highly predictive models. And it's important because you still have this sort of physical understanding, but now you sort of taken it to a place using AI that using SilCS alone can't get you. We're getting into the like drug liability, and this is in the context of H-erg blockade, where we basically went in, we calculate the FragMaps for the transporter. So this is just an example of how we can start to apply the FragMaps to very large complex systems. And then we combined the FragMaps, we applied our Bayesian machine learning optimization with a known database of H-erg blockers, develop a predictive model, which we subsequently have improved. And again, sort of now including the SilCS FragMap LGFE scores along with physicochemical properties. So again, this gets to a combined SilCS machine learning sort of combination to develop predictive models, which you can use to sort of understand potential for H-erg blockades of molecules as you're developing and so on. And again, you still have this sort of, you have something that's is blocking H-erg, you can understand what parts of the molecule are contributing to that and so on. So again, this information content's very high. We can do protein-protein interactions by docking one protein with another protein in the context of the SilCS FragMaps and probability distributions of the functional groups in the protein. It's nice because it gives you the full distribution of potential binding sites, and we show that it reproduces known binding orientations and so on. And this has been interesting because it's now allowed us to move into the area of biologics formulation, where what we want to do is sort of use SilCS to predict excipients that can be used to stabilize proteins, as well as limit protein-protein interactions, which lead to increased viscosity and aggregation. And again, the SilCS is very good for this because it's mapping the entire three-dimensional surface of the protein. We can identify where all the excipients bind. We can identify where protein-protein interactions have a potential for occurring, combine all that information to understand which excipients have the best potential for limiting protein-protein interactions and so on. And what was interesting, and this is a more interesting study, which has just come out in Molecular Pharmaceutics, where we can actually, you take a protein, you can calculate the charge of a protein based on just the pKa, but what's relevant is the charge taking into account the buffer conditions, which means like what ions are actually stably bound, what buffer molecules are bound, to get the actual effective charge of a protein, taking into account the environmental contributions. And we can show that we can use this to get a very highly predictive model of, so when you change your solution conditions, the charge of the protein changes. And if you're doing biologics formulation, you want your protein to have a large charge so it repulses itself and so on. So again, this is a very powerful approach. And then we've also using it for basically understanding intermolecular interactions, where we're looking at ensembles. We generate large ensembles of interactions between, say, these would be like ITAMs and kinases and so on. So we're using it in the context of this is more sort of application approaches to understand sort of signaling and so on. And you can really, and the thing is with the SilCS technologies, you can generate huge ensembles of conformations, 30,000 conformations. You can then use SilCS for comprehensive docking to generate, almost 500,000 poses of the ITAMs interacting with LCK. Then based on sort of overlap, trim that down. And basically you can really start to understand these sort of like as receptors come together, how it increases the potential for binding to like a kinase. And I'm obviously going through this very quickly, but it again, it speaks to the utility of the SilCS technology. And then finally, we can also do SilCS in the context of lipid bilayers to understand the distribution of functional groups across the bilayer. And then once we've got these functional groups across the bilayer, we can now start to use it to understand the free energy profiles for ligands passing through a bilayer, which now allows us to get to models to predict passive permeability. And again, now what we've done is we've taken the SILCS FragMaps, we calculate the free energy profile of a ligand going across a bilayer. We've now combined that with deep neural nets to develop predictive models of ligand permeability. And again, these are quite predictive models, and they seem to be better than anything that we've seen that's out there right now. Because SILCS is so fast, you can actually explicitly take into account the neutral and the charge state and make combined free energy surfaces and so on. And again, we can then understand which moieties in the molecule are contributing to the barriers to permeability and so on. This is sort of a whole other realm of the SILCS technology and so on. And then I just want to emphasize that we use the CGNFF program to just generate all of the topologies of parameters and then input that into the SILCS calculations. So basically sort of the topology parameters, it all just disappears. It's just all automated, and you do your SILCS calculations and get your results, and then you just regenerate all of that in the future if you move it. And we're now sort of moving towards SILCS as a function of pH, using SILCS in the presence of water, looking at SILCS for ligand off rates. And we've got our polarizable force field, and we're thinking about moving SILCS towards using that and so on. And this is interesting because polarizable force field's slower, but once you've got the frag maps, they're going to be the same speed as what we're doing now and so on. And then just to acknowledge the large collection of people over the years who have contributed to the development of the SILCS technology. And here's my current lab members. And thank you.
Thanks a lot, Alex, for a very comprehensive overview of SILCS and its many applications. We have time for questions. So if you have not yet typed your questions, please do so. We will read them aloud. We will read them to Alex, who can then answer. Sarah, do you want to do that?
Yeah,
please?
We can get started. So the first question was from Michael, and this was from the slide where you were looking at the frag map of PTP1B. He said there were several sites where it looked like multiple function groups could have affinity.
Yes.
And so the question is, how do you determine which of those is a good drug binding pocket or which is really the druggable site?
These are different maps, but you can see here you've got basically aliphatic, aromatic, which is not unlikely, but then you have donor map, or excuse me, hydrobond, acceptor maps all on top of each other. The point here is that because the maps take into account desolvation effects as well as protein functional group interactions, this is telling us that any of these functional groups could be placed at this location. And this is the dimensionality of the information in the frag maps is such that it's taking into account all of these effects. There's something that is not just a hydrophobic site, but there's enough polar interactions that you could put an acceptor there or a donor there, as well as a an aromatic. And there's also like a heteroatom aromatic becomes obvious. So you basically you can put both there. That's what it's telling you.
Okay, Sarah, do we want to move to the next question, please?
Okay. So NATO is asking, how do you handle protein conformational changes and fluctuations when creating the frag map? So like if part of the protein moves from one voxel to another,
well, this is actually a good
disturbed that you're getting averaging over different bits?
Yeah, what we're doing is when we do the SILCS simulations, we have very weak harmonic restraints in the C alpha atoms. So we don't get a big conformational change like from an active to an inactive form, but we're getting the local flexibility. And so this is how we go from, like, this is a solvent accessible surface. So the amount that the protein can shift its local conformation is taken into account. So this is why you see here how these maps are actually penetrating underneath the surface. So as the protein can open, as it samples its local conformational space, that's taken into account in the FragMaps. And this is what this is telling us. This region of the protein can actually relax when we bring in a new ligand, or if we add a larger functional group to a ligand, the protein can open and we can add a functional group underneath the surface from a crystal structure because the protein has that flexibility. So that information is inherently in there. This is why when we get to using the SILCS for the ligand design, we get rid of the protein structure because it's a one snapshot and it doesn't really account for the protein flexibility. The SILCS FragMaps has that information.
Okay, and does that kind of wiggling cause noise and blurriness in where the fragments are relative to the protein?
Well, what it does is if you look here, you can see like these, you can see these regions have opened like around here and over here. And what typically, because the solutes are competing with the protein, so the protein occupies that region some of the time, the solutes occupy some of the time. So your FragMaps are less favorable, okay? And if the protein wants to be there most of the time, and the FragMaps very little of the time, then the FragMaps in that region will actually be unfavorable. So that is inherently included in the maps. So how much it can compete with the protein conformational flexibility and so on.
Okay, so yeah, but that's a very interesting, I suppose, clarification. So the protein does not diffuse, right? It's somehow held in place apart from local fluctuations. Because otherwise, if your protein were to diffuse, it would be maybe practically quite complicated to reassemble the whole FragMaps.
Yeah, technically you can deal with that, but the issue is like like the question was sort of saying, it's like, well, you wash out these FragMaps, right? So we want to stay in like an active conformation versus an inactive conformation. So we want to sort of sample the local conformational flexibility. But if we had, say, a big loop that sort of moved way out of the way and we sort of had all of that space, then the FragMaps would be totally washed out, okay? So we stay in the local region of conformational space, take this into account. So this is giving the information to facilitate our ligand design to get rid of the local conformational rigidity of the protein. But at the same time, we don't wash the FragMaps out so much that they become like sort of meaningless. So if you have like two conformations of a kinase, GPCR, you want to do two separate maps. Okay.
Right. As a further questions around, maybe not right now, people can continue to do not hesitate to ask questions. I have a couple, in fact, since I will take this opportunity to ask some questions as well. So you, in your system, in the solvent, in the bulk solvent, you have what seems to be quite a high concentration of diverse fragments. You mentioned 0.25 molar for 0.25 molar. I suppose it's per fragment type. Is that are these high concentrations sometimes a concern, or has this been, I suppose, a lot of work went into choosing these concentrations?
A lot of work. We started with like just a couple solutes at like 1 molar, and we're getting pretty good behavior. And then we went to 0.25, and it's worked well. One of the things that we do is we have repulsive potentials against between, say, benzene with itself or benzene with propane, so that you don't get hydrophobic aggregation of those.
Okay.
and we have repulsive potentials between, say, methyl ammonium and acetate. So we sort of like enforce like a little bit of an ideal solution. Now, and those repulsive potentials are over a fairly short distance, like less than 8 angstroms. So we avoid the aggregation problem or the, this, ion pairing and so on.
Okay, now that's another very interesting explanation. So there are a few tricks that one needs to use, but that's so that's really interesting. I was also wondering, when you work with membrane proteins, presumably there's an explicit membrane as well.
Yes.
And if so, do you see some of this? Do you see some artifactual maps, let's say in the membrane itself, or in places which are not particularly helpful? Does the membrane itself give you some maps,
Well, this is, I mean, we initially were like trying it with a GPCR, and I don't think, I guess I have it. So we're sampling solutes throughout the bilayer as well as in solution, as well as inside the protein. But you get these probability distributions in the bilayer, which are totally physically realistic. If you go back to our paper, but this point here, this is just doing SILCS in a bilayer. And so, yeah, you've got these distributions out in the bilayer, okay? And maybe that's not going to help for your ligand design, although maybe if your ligand's coming in and through the bilayer, it could be useful. But it's interesting, you have then maps on the surface, say, of your GPCR. Often they are more hydrophobic, or as you would expect, but you're getting, but you can see regions. It's interesting, in the mu opioid receptor, there's a binding site for cholesterol, and it really binds there tightly. And when we did the SilCS simulations one time, I removed that. And because our simulation system is POPC with 10% cholesterol, basically, during the SilCS simulation, cholesterol went back into that site, and there was no maps in that site because the cholesterol just was so effective, competed them away. So you're getting very favorable sampling. I mean, this is these functional group distributions that we're getting are very representative of like the energetics of how functional groups sample environments, be it in a bilayer, be it, on the surface of proteins, be it in these binding pockets, on RNA and so on. It's interesting, and that appears to be a big driver for molecular interactions as we'd expect.
Yeah, no, so one thing that I really like about that is that it has a good degree of interpretability, right? Which not all techniques have. So for instance, with FEP, people complain about that. Although here one can, I think you get some good indication potentially as to why why certain groups want to bind. So, and in terms of extracting semi-qualitative information, which would be very interesting to a drug designer, do you, for instance, can you quantify, or can you give some descriptors, where can SILCS give some descriptors in terms of desolvation? When you look at your system, can you say, oh, it's clear that this pocket is more easily dissolvated than another, or does that need an extra level of analysis, or is that immediately visible from the FragMaps?
That's pretty immediate. This is when I showed these pictures with water, is because you get the you're getting the distributions of water. And obviously we're looking at these as a certain cutoff. This is like, and you can see which regions where water binds tightly, okay? And then you can see where the FragMaps, like you see, look at this site. You see that you have FragMaps competing with that, okay? Now, you look down at this site, no FragMaps compete with that. So that's telling us where you can compete with water effectively and where you can't compete with water. So that information is inherently in the maps. Right.
Okay. Yes. So, Sarah, are there other questions? Has a question come or?
All quiet.
People are too shy today. Sarah, do you have further questions?
No.
Yeah, so I have in fact one final question. So we saw several examples where there was a first fragment and first compound, and it was modified, and affinity was gained at one end of the new compound, and affinity was lost at the other end. And I think there was more than one example of that. That's intriguing. Is it because when forming a stronger interaction at one end of the compound, one shifts the compounds, which then disrupts the interaction at the other end, or is there another?
Yeah, pretty much, yeah. And right, so that gives you the information. It's like, okay, do I increase the linker length so I can maintain binding at both ends?
Okay.
Yeah. That information content it's incredibly high. It really takes people a long time to wrap their head around all the information that's coming out that you have accessible to you and so on. And then again, the quantitative accuracy, it's like it's there with FEP. It really speaks to, it's interesting because it's saying that, FEP, like SILCS will never get like the absolutely correct binding affinity, right? Because we have this, we have the precomputed ensemble, and FEP in principle will, right? However, FEP and SILCS give you basically the same predictability. And it's just telling you that what's really the dominant driving force is just the functional group affinity, which we're now taking into account the entropic contributions, desolvation, and all these types of things. It's just, that's sort of, what's really the driver. SILCS is like an experiment and just, okay, what's the most important thing for predicting binding affinity? And we don't even take into account ligand conformational strain or anything.
Yes, that's interesting as well.
We do the docking. We are using the CGNFF intramolecular energy, so you don't sample like unphysical conformations, high energy conformations. But the final LGFE score doesn't have like a conformational strain contribution.
Right. Yes. It's in fact the same was somehow true in the very early LIE calculations, which go back now a good 20 years ago, and yet they managed to get good correlations. So that can be a bit intriguing. I don't think that I would have further questions. I learned a lot. That is going to be the last call for more questions. If you really want to, ask your questions, you can unmute yourself, and if you don't want to write, you can unmute yourself and ask your question directly. If that works better for some people. If not, if not, I think we will thank Alex again for a very lively presentation and some very, very interesting examples. And now SILCS is even helping with excipients. What's next?
These things I never sort of imagined when we first started this, but it's
yes. Yeah. Okay, well, I greatly enjoyed it.